Dr. Jack A. Gilbert is a professor of microbial oceanography in the Center for Marine Biotechnology and Biomedicine at Scripps Institute of Oceanography and holds a joint appointment in the Department of Pediatrics in UCSD School of Medicine. Dr. Gilbert is also co-founder of the Earth Microbiome Project and American Gut Project, has authored more than 350 peer-reviewed publications and book chapters on microbial ecology, and is the founding editor-in-chief of M Systems Journal. He has been recognized on Crain's Business Chicago's 40 Under 40 list, listed as one of the 50 most influential scientists by Business Insider, and was named as one of the Brilliant 10 by Popular Scientist. He is the co-author of Dirt is Good, a popular science guide to the microbiome and children's health. Dr. Sean Gibbons is assistant professor at ISB. He holds a PhD in biophysical sciences from the University of Chicago. His graduate work focused on using microbial communities as empirical models for testing ecological theory. Gibbons completed his postdoctoral training in Eric Elms' laboratory in the Department of Biological Engineering at MIT, where his work focused on developing techniques to quantify individual-specific evolutionary dynamics within the human gut microbiome. He is particularly interested in learning how organisms in the human gut change and adapt to individual people over their lifespans and how those changes impact health. Please join me in welcoming Dr. Jack Gilbert and Dr. Sean Gibbons.
Thank you, Faith. I'm very excited tonight. Like Faith said, my name is Sean Gibbons here at the Institute for Systems Biology. I'll give you a little bit of background on ISB before we jump into it. So ISB was started in 2000 by genomics pioneer Dr. Lee Hood. We're a nonprofit biomedical research institute located in Seattle's South Lake Union neighborhood. And we work on some of the most pressing issues in health, including the microbiome, aging, COVID-19, brain health, cancer, and much more. There are two ways you can learn more about ISB. You can visit our website, isbscience.org, all one word, as you can see over here. This next week, actually, we have our annual Reimagine event coming up. And this year is an exciting year. We have this conversation scheduled between Dr. Jim Heath, our president, and Nobel laureate Dr. David Baltimore, and an immunology expert, Dr. Mark Davis. And they'll be discussing the immune system, and in particular in light of this pandemic we've just been living through, and the sort of research emerging from that. So you can, again, register for that event if you'd like at isbscience.org. And I'd like to express our thanks to our partner, Town Hall Seattle. For the past decade, ISB and Town Hall have been putting on a number of great science-focused events. We look forward to putting on many more. And again, if you have questions for Jack, please add them in the chat. Okay, I am really excited to introduce Dr. Jack Gilbert. I won't go through all the things that were said by Faith. She gave a pretty great intro of all his accolades. I would say Jack is a microbial ecologist by training. His lab uses molecular techniques to study microbial communities and how they behave and function. We go way back. I was advised by Jack. He was my PhD advisor along with Maureen Coleman when I was at the University of Chicago. He's been at the forefront of microbiome research for years and a real champion of science communication, I would say. I learned everything I know along those lines from him. And that's why I wanted to chat with him today about the current state of the human microbiome field. Where did it start? Where is it now? And where is it going? Let's see one last thing. It's a pleasure. Okay, I think I said everything I need to say. Welcome, Jack. It's great to have you here.
Thanks, Sean. Yeah, you were actually my first graduate student in the United States, which is
yeah.
I know, right? It went all right. You turned out good.
Yeah, you did a good job. Yeah.
I don't know. I did nothing. I just gave you some pointing guidelines, I think, and you did it all yourself.
It awesome. I remember you showing up at UChicago and just being this, force of charisma, and very inspired to join up with you and work on a bunch of cool projects. Yeah, so I guess, let's jump into some questions. I have some sort of strange questions for you at the onset before we jump too far into microbiomes. So I have a bit of a passion for the confluence of art and science. I come from like a background of artists myself. I know that you are a musician, and I think you had a band when you were younger.
Yeah,
Um,
when I was older as well.
you still play, right? Yeah.
Yeah.
So, thinking about that, how do you see that having informed you as a scientist? And do you think there's value in having art in your life from the perspective of a more quantitative scientific person?
Man, yeah, I'm married to an artist, so yeah, I definitely think it's important, right? And I love your dad's poetry, by the way. Your dad's a phenomenal poet. I think he's absolutely splendid. If you anyone out there is interested, you should look up Mark Gibbons. He does incredible poetry. Yeah, I mean, art to me is the same side of the coin to science, right? They're both highly imaginative fields that require dedication, training, experience, and a desire to practice continuously. I've not really. My parents weren't particularly musical, but my kids are both musical, and my wife and I play music every day. And I think practicing music is one of those things where the more you do it, the easier it becomes. The more you find out about it, the more you get involved with it. And science is the same way, right? And it's quite remarkable how many scientists, especially in academia, but also in industry, and I've worked in both those fields, are musicians, right? Or painters, or poets, or some other form of self-expression. And I think self-expression is absolutely central to being human. So art is just another side of humanity. But as you say, it's a phenomenal opportunity to explore who you are and what you can be. And so I feel that science should be the same, and I think they're intrinsically linked.
Yeah, no, I agree with you that I think the best scientists tend to also have some sort of artistic expression in their lives. Although I would say the culture of science doesn't necessarily support that, right? I mean, I don't know. I wouldn't say that's the truth of the vast majority of people practicing science, of having a sort of artistic hobby,
Yeah,
and even
I
poo-pooed a
think
little bit, right? By some
yeah, I
science.
think especially in America, right? There's a significant push towards working as hard as you phenomenally can, never taking a holiday, and never always being available, and that doesn't really leave a lot of time for you. I've always tried to. I think everybody thinks I work extraordinarily hard, but I think I like to think I work smart because I always try and carve out time for my own mental health, for my own sanity, to do things that give me pleasure, right? And in my lab we have a code of conduct that everybody has to read, and evolves over time. People add to it, but there's a certain degree of adherence that explains that everybody is who they are and who they can be and want to be, and we should respect that, right? And give people the space to be human
Yeah,
as well as to contribute to wonderful science. And I think those two things aren't mutually exclusive. There is an opportunity to redefine the work ethic that we have to free up time for those personal pursuits which give you pleasure, right? And give yourself the breathing space to also be human, which is especially as an early career scientist in your field, you must feel that pressure in academia, especially in I don't know what it's like up in ISB, but universities generally, yeah, we put so much pressure on people to perform and to be on all the time. It's brutal, and then we question why there's severe mental health crisis in academia. Something needs to change. It needs to change now, and I'm trying to do what I can in my space, but there needs to be a revolution.
yeah, yeah. No, I think we need to take the time to be human, like you say.
Yeah.
Um, all right, so moving into your kind of career and how you got into what you're doing now. As far as I remember, you started off chasing butterflies early
Yeah.
on. So I guess you were an ecologist at heart always. But what transitioned you into microbes?
I'm a magpie or a jackdaw. I just see shiny things and keep on going for them. Yeah. So the I was I first time I saw a microbiology anything was a textbook that I got that was delivered to my house in my first year of university, which was quite remarkable because I nearly failed high school, the equivalent of high school in the UK, my A levels. And so, I basically scraped my way into what at the time was free university education in the United Kingdom. That was subsequently scrapped. Now people have to pay for it, but and managed to find a space that I felt comfortable in, that I felt that I could apply myself to, and that was remarkable for me. I felt super excited about it, and I was never that good at exams, but what I really was good at was I found was working on a problem and being tenacious at it, right, and trying to find it out, because I found that I was passionate about just nitpicking and digging away at problems until we came up with solutions. And so I started working at the Natural History Museum in my second year, and there's only 3 years in a degree in the UK at that time, and managed to get hooked into trying to figure out how to model butterfly distributions. I mean, of all things, I had no idea what I was doing. I still don't. And that because it kind of spiraled out of control. I remember I was in a lunch break after sitting down and cataloging what must be my 15,000th butterfly from the 19th century collections in this dusty old room, and sitting down just grabbing a bite to eat outside, and somebody gave me a called my phone and offered me a PhD, but I had to go and learn microbiology and work in Antarctica for 2 years, and I was like, sure, that sounds like fun. Let's do that, and that's pretty much been my raison d'etre ever since. If something sounds exciting, it probably is, and you should at least explore it.
Yeah, I feel like that's a good recipe for success in any sort of professional field that's really competitive, is grabbing those opportunities as they come and trying to engineer it from the beginning. I was never good at that either. Yeah. All right. Well, let's get into microbiomes then. So from your perspective, what is a microbiome, and why do you think it's important?
Well, what is a microbiome? Well, microbiome is what we used to call microbial ecology, and which essentially it still is. It's just an ecosystem of microbes, right? Anything that's small. And that could be eukaryotes, protists, little fungi, anything that's single cellular normally, and viruses, bacteria, archaea, the whole gamut. And to me, I like to think of it as a living, breathing unit of life, right? And you find it everywhere, and that's the beautiful thing about life, is it finds a way, as Michael Crichton said. These are organisms that have been around for 3.5 to 4 billion years, and they've colonized every facet of this world in unique ways. And the field of the human microbiome really has only come along in the last 20 years, and but we're just another lump of flesh that microbes have colonized, and we've had to adapt our entire bodies, our entire body plan, our immune system is all just adaptations to dealing with living in a microbial world, of being exposed to microbes all the time. And when you see it from that perspective, it really does change your understanding of the world we live in. Everything from the pioneering work by Margaret McFall-Nye and Ned Ruby on Vibrio relationships with cephalopods, all the way through to trying to understand what really drives plant adaptations, as you and I have done in the past, right, with your work up in Montana, in different environments, and seeing how they actually function. Once you realize that everything that we can see with our eyes is just trying to survive in a microbial world and take advantage of microbes where they can and steer away from them where they can't, it really changes your perspective on the biology of our planet. And so for me, that's always been a driving force, to really try and see the world from a microbial perspective and understand what it means to swim in that soup.
Yeah. I mean, I feel like all of sort of 19th and 20th century microbiology was about the bad guys, right? Studying the pathogens, and so microbes, bugs, people had like a negative connotation with them. but I feel like the microbiome field has been a paradigm shift in how we think about it, because they're not all bad guys, right?
Yeah, well, I mean, it's funny. Even you go back into the late 19th century, it's not like everybody always thought they were all bad guys. I mean, even Louis Pasteur said that he didn't believe that certain animals or plants would be able to survive without microbes, right? So people knew it. People instinctively knew that there were, these things were living organisms with their own game plan, but their adaptations were manifold. It wasn't like, it's not even like right now, if you take Yersinia pestis, it's not like Yersinia pestis' entire game plan is to infect people and kill them with bubonic plague, right? There are other facets to its character. I like to say this, it's not like everybody that goes to jail is a criminal, right? There are just people who got into the wrong circumstances and, due to the mind-numbing and degrading poverty that many Americans have to live with, got into the wrong situation and had to take advantage of that situation when it happened. Bacteria are the same. You can demonize them all you want, but to be perfectly honest, they're just trying to survive in their world as well, right?
Right.
Freedom.
And if your survival helps them, then they're going to help you, right? They're not necessarily good guys, not necessarily bad guys. Context is key.
Context is everything. Yeah.
And this leads me to the next question, because I know you've published a paper on this very topic, but how would the world be different if all the microbes just disappeared overnight?
Yeah, so this was Josh Neufeld's idea. I think we were having a beer in some place in Colorado, and he said, I think this would be a good idea. So we just sat down and postulated, how important are microorganisms on the planet? And it turns out when you do the thought experiment, they're pretty important, right? Now, like, what do you call a microbe? If you take away all the mitochondria and the chloroplasts, which are ancient microbes from our bodies, then everything drops dead instantly, right? But if you look at, say, bacterial nitrogen fixation, there are maybe a couple of new reports of some eukaryotes that have evolved certain mechanisms that are akin to nitrogen fixation, but aside from that, 99% of all the nitrogen on the planet that's not fixed by human processes is fixed by bacteria. Imagine taking that away. What happens to all the plant life on planet Earth if suddenly you start to run out of the available nitrogen stocks? You need nitrogen for all of our processes, for all of our proteins, all of our DNA. It's kind of written there in the code, right? Nitrogen is important, and that's just one facet. They are central to everything on planet Earth because they were here first. I mean, they're the one that made this planet oxygenic, right? Pumping tons of oxygen. Every other breath you breathe comes from microorganisms pumping out oxygen from the ocean, right?
Every other breath comes from chloroplasts that are ancient microbes enslaved by plants.
I like that.
Yeah,
that's a good one. I'm going to steal that. I'm going to use that. Right? It's really all microbes. All these little fleshy things walking around thinking they're all that. It's kind of nonsense. Yeah, but I do love that idea, right? I love doing thought experiments, and again, harking back to science, that's what science is. Sit down and think out the problem. Work it through. If you can do that, and especially if you have really, engaged people to work with, right? Like you and I used to sit down and do this, and I still do this all the time. I try and sit down and go, okay, what is the actual problem? Let's work it through. Figure out these experiments. Try and think about what the experiment's going to tell us. Try and think about what other experiments we should be running, and realistically, what's the end goal? Why are we doing this science? And so those thought experiments become incredibly important in helping to shape what you think you can do, what you can apply yourself to in order to find the next discoveries that are incredibly important.
Yeah. And I'm sure these thought experiments drove you to some of your bigger projects, right? So let's talk about the Earth Microbiome Project. How did that emerge and how did that start and what was the impetus for it?
Yeah. I mean, we've done this large scale study in the United Kingdom, looking inspired by Jed Furman's work from University of Southern California and Jen Martini's work. She's now at University of Irvine, UC Irvine. That were exploring how microorganisms fluctuated over time and space, and I'm still obsessed with this. Right, what is it about niche space, the n-dimensional hypervolume, I love that term, of environmental parameters that shape when and why a microorganism can grow and breathe and live, right? And that to me has always been fascinating. So understanding how things change through time became incredibly important to me. And I quickly realized I was unable, or it was impossible to study microorganisms in the context we wanted to study them in one environment, let alone trying to see if patterns that we observed in one place were propagated around the world, right? I always wanted to find that universal rule, like the thing which drives everything. And in the English Channel, it looked like microbial life was driven by the amount of daylight that was available, not due to cloud cover or like sun intensity, just the length of the day. It gets dark out here now because the sun's not hitting San Diego as much as it was in the summer, right? And that length of day seems to be a vital clue to what drives the energetics of ecosystems. So I wanted to see if that was propagated around the world. And so we got the great and the good, right? We got Rob Knight, who had already been exploring tons of microbiomes around the world and was, already set up to try and explore this. And a bunch of other scientists, Ashley Shade, who is currently a phenomenal assistant professor in MSU and doing some incredibly important work. And we got them all in a room and we said, all right, let's do a thought experiment. If you had unlimited budgets, what would you do to try and catalog microbial diversity on Earth and find these patterns? What would be the driving force behind that? I mean, why would you do it? How would you do it? And realistically, what are the gaps? What are our things that we need to overcome in order to actually achieve it? And out of that came, what became the Earth Microbiome Project. We got a very talented graphic designer called Eamon Maguire, who was already working for us in the Genomic Standards Consortium to do a freebie and come up with a logo. Once you got a logo, you got everything, right? Then it all just, carries on from there. And, I sank all of my startup capital from my, I moved to University of Chicago and Argonne National Lab, and all my startup went into actually running that project. And, Rob put a ton of his money in, and Janet Jansen put a ton of her money in, and we all kind of tried to see if we could collaborate with scientists around the world to get a load of samples in and just start sequencing. And that's all it was, right? And you ended up with, well over 500 collaborators. Everybody put their 2 pennies in. Everyone had their own little research project they wanted sequencing for, and we just enabled that. Developed a whole ton of kick-ass, programs and tools and protocols, which are now industry standard, right? The protocols the Earth Microbiome Project developed are what everybody mostly uses. And it enabled us to create this one giant database, which sounds lame, right? Who wants a big giant database? But with giant databases, you can ask really important questions. And we're still asking and answering those questions to this day. It's never really ended. It's just continually trundling along like some kind of out of control train.
I mean, I remember being struck as a student by the audacity of the project, right? We're going to sequence the microbiome of the Earth, of everything on Earth. The Human Microbiome Project was just like 300 people, but you were going to do everything.
Boring. Yeah. Well, those 300 people had a lot more money, spent on them than we had to spend on the planet. But I think that, when you're scrappy and you don't get all the money in the world, you figure out ways to do things that change the world, right? I honestly believe that still. I think some of the projects where we're most innovative are the ones that are not funded, because we have to scrimp and scrape, save every corner and come up with innovations that will actually help us to get that project done. When someone drops a ton of money on us, oftentimes it causes more problems. I'm not saying that out loud, am I? Oh Christ, someone's going to have a go at me, like the NIH is going to come after me for their money back. But I think it's important that you realize, right, that when you have a vision, you find the people that you can work with to make that vision possible, and the people that share the passion, right? It's not always the people that are best at it, it's the people that share the passion. And if you all share the same passion, man, then you can do anything. can change the world and change the ecosystem entirely. And I really honestly do believe that's what the Earth Microbiome Project did. It laid a foundation to set up what has become a field which has changed the world. And that may be hyperbolic.
Do you have like a favorite biological insight or two that's come out of that work? Is there something that you think is?
I mean, the thing that I think is and I think that this your PNAS paper from 2012, I think, was a fundamental start of this. But my obsession with the fact that everything has the ability to be everywhere, right? Distribution of microbial life is constant. And what the Earth Microbiome Project proved is that is almost a reality, right? If you look in any environment on Earth, you find the same phylogenetic backbone of microbial life being generated and then selected for by very selective environments. You take a hydrothermal vent, you've got 120 degrees centigrade water spewing out of the ground, 4,000 meters under the ocean in the pitch darkness, covered in sulfur, and yet these things are ephemeral. They just pop up out of nowhere, and suddenly all the microorganisms that are super adapted to that environment appear. Yeah, right. Judy Huber, a phenomenal microbiome scientist and working at Woods Hole Oceanographic Institute, has done some really cool work looking at how these bacteria come out of the fluids in the earth, but they're also coming out of the ocean themselves, and we find them everywhere, right? We find them distributed around the world, just incredibly low abundances. And that to me is the most exciting thing, the fact that the microbes in my body came from somewhere. They didn't just sporadically appear. They are colonizing me. And a lot of them are passed down, from person to person. We just had a paper published with Ram Bleckman and a lot of other collaborators. Beth Archie, and everyone. A phenomenal group of scientists. I'm just so lucky to be even associated with them. But showing that baboons share this rich, diverse microbial flora within their tribe, within their pack. I can't remember what the collective noun for baboons is, but and that that 90% plus of that microbial community is heritable, right? It shares it shares the same trend for kinship as that population does, and that's phenomenal, right? It says that there is sharing within that population, but those microorganisms are also coming into that population and adapting through it, and I think that's beautiful.
Yeah, and it takes us to so you've done a lot of work in the built environment. Speaking of baboons being colonized by their neighbors and by their environment, they're out in the wild and in the forest or in the plains. Human beings are in a very different environment. So can you talk a little bit about how you think the built environment has affected the human microbiome?
Yeah, this is a difficult one, right? The way I would have talked about this 10 years ago is probably very different to the way I talk about it now. In fact, when a program officer invited me to get involved with it at the behest of Volker Meyer, who was one of my colleagues at Argonne National Lab, I honestly thought it was kind of nonsense, right? There's not a hell of a lot of microbial diversity in this, but we got some money, and if someone gives me some money, I'll do something with it, right? So we had a look-see, and it actually turned out to be some pretty interesting trends. I could do some cool ecology in an environment where there's very little life. And I know your side of it. The whole thing's a desert and everything's dead, right? Yeah, but even deserts are ecosystems.
Oh, I know. I totally. I mean, I do think that things are like dead and dying on a lot of these surfaces, but they're also reservoirs, and in a sense, the fact that they're dead surfaces is very different from if you're in a forest living your life, right? So that's right. The potential for us to be colonized by diversity is perhaps dampened and.
But remember, we're not always being colonized. I think the major impact, to answer your first question and to spin off of that I think we're feeling is a lack of antigenic exposure, right? Our bodies are used to being constantly bombarded with antigens, and work we did with Carol Ober and Anne Sperling and Donato Bocelli and other colleagues at Chicago and Arizona on the Amish and Hutterites really demonstrated that, right? When kids grow up in a farm and they actually have the opportunity to live and work on the farm, you can see the signature of that in their immune systems. Their their immune profile is reflective of being constantly bombarded by things, by antigens, by bacteria and viruses and fungi and dirt, right? And that to me is a key precept, that our bodies are expecting to see that kind of stuff, right? And it spins off of the hygiene hypothesis and the old friends hypothesis, the fact that in history we descend from people who would have been exposed to lots of things, and the fact that we're not exposed to them but now is making our immune systems dysfunctional. Like they can't quite figure out how to respond properly to the antigens when they do appear. And I think that's a key precept of what's missing from most of our built environments. Right, when kids have a dog in the home that they can physically interact with, they have a 13% reduction in likelihood of developing asthma. I mean, these epidemiological studies demonstrate there's an importance to that exposure. And so, from my perspective, I've always been about, well, I want to figure out the bidirectional mode of interaction between us and that environment. And that's actually led me down some really weird paths, right? Now we are actually adding bacteria back into hospitals. We're adding bacteria into our homes in order to see if we can not just stimulate immune responses. We're not there yet, although there's some really, really cool work coming out with some collaborators in Finland soon that is exploring that phenomenon. But we actually think that having bacteria in the building can actually reduce the spread of disease. A lot of this comes from the fact that when the bacteria leave our moist, warm, cozy body and end up on these cold, harsh, dry, desert-like surfaces, right? Most of them die, as you pointed out. But the ones that survive are like Mad Max hardcore survivors, right? They hang around, and they often have these adapted genes which make them dangerous to humans, more virulence, more antibiotic resistance. But not if you chuck a load of bugs in there that stop them from growing. And that's what I think is so cool, right?
Yeah.
You give them a forest. You can't throw a house plant into a rainforest and expect it to grow. These bugs that leave us, they're only surviving there because there's nothing there to outcompete them. So we're giving them competition. And I think that will be the frontier for changing built environment microbiology for good.
Pollution is the solution to pollution.
All righty.
This is like a hospital probiotic you guys are working on to try to
dampen
antimicrobial resistance. And
well, again, a brilliant group of people. Actually, Megan Timmis, who's one of our postdocs, she's working on this in hospitals in the San Diego County area, who I won't name, but also working on a similar program with NASA. And actually, Megan MacArthur, who's an astronaut for NASA and also a Scripps Institution Oceanography alum, has been doing the sampling for our research projects on the International Space Station, exploring these types of phenomena, which I think is so cool. It's all connected.
Oh, it's super cool. And I guess that also, let's see, I'll try to scale back some of my questions. We're getting close to audience questions, but I have a few more I want to get to.
I did say, you get me talking. You can't stop me from talking.
The time flies when you're having fun. Well, I guess, we sort of covered all this, but, you wrote this popular science book with Rob Knight, right? Called Dirt is Good. And just maybe briefly.
I didn't name it.
Yeah.
That wasn't our title. Yeah, I mean, it's the precept that being exposed to things is important. Rob and I are both new parents at the time, right? And in fact, our children are born on the same day, which is kind of weird. I know we've always been intrinsically, strangely linked, but in the context of this, we just wanted to provide a right kind of advice, right? There's a lot of nonsense out there, and you can get any kind of advice you want on the internet. So we just wanted to take all the knowledge that we had and our collaborators had and just put it in a way that could actually be translated. We found this wonderful writer called Sandra Blakesley, who helped to translate some of our sciences into less dense language. But I'm proud of that book because the way we were able to present it is still accurate, right? There's nothing in there which is hyperbolic. There's nothing in there which is inaccurate. There are accurate statements, and I like accurate statements. It means that you can trust the information in the book too. If you're interested in raising children, it's a cool thing to do.
Yeah, it's just an hygiene hypothesis in general is an interesting area. we definitely have seen improvements in human life from hygiene, right? Like that, infectious disease is
enormous ones.
Yeah,
vaccines, hygiene, they're important.
but there's a huge cost as well that we didn't recognize, which is this disruption to our immune systems and the rise of these inflammatory conditions and the sort of developed world syndromes that we suffer from now. So how do you strike that right balance? And I think that book kind of gets into that. I couldn't resist. We were talking about some of the ongoing projects that you have in the lab, and I heard about dolphin FMTs, fecal transplants for inflammatory bowel disease. Dolphin inflammatory bowel disease.
Yeah, man, they get to eat some bad stuff when they're out there in the ocean. So these are dolphins that are wild dolphins, that domesticated wild dolphins. They live out in the wild, but they are friendly to their handlers. Leave it at that. And yeah, and some of them get severe IBS and IBD, and so we're interested in exploring how to manage their colitis symptoms when they do get it. And so we've been doing fecal microbiome transplants from healthy dolphins to non-healthy dolphins and seeing if we can alleviate the symptoms. And it appears to work quite well. Cool. And if you think about it, a dolphin's just, a very friendly and very much cleverer sea dog. So, they're just a wonderful animals to work with. They have personalities all their own.
Yeah. That's awesome. And I guess before we run into the other questions, I'll just kind of leave off on my questions with, when do you think everyday people out there listening in the audience, for example, are going to start to see the impacts of the human microbiome field in their daily lives? When are they going to have access to this sort of research in clinical medicine and all these other things?
Yeah, other than FMT, fecal microbiome transplant for treating recurrent C. diff, there are no FDA-approved therapies. There's one that's extremely close with Seres Health. That's another, targeted therapeutic for treating C. difficile infections. So everything is C. diff, C. diff, C. diff, C. diff. And yet, the public can access them. And I'm on the scientific advisory board for Day 2, for example. I have to announce that. This is work that came out of the Weizmann Institute with Eran Edelnoff and Eran Segal. And Eran Segal's an incredible scientist and bioinformatician who's generated algorithms that capture a lot of information and hunt down traits that can be used to predict how to drag people out of pre-diabetes into diabetes. And I begged, borrowed, and stole my way into that company. And my dad used the company to bring himself out of a pre-diabetic state. So he managed to stave off type 2 diabetes and is now much healthier than he was. And because the algorithm predicts a diet, right, that is effectively personalized for him. Yeah, exactly. To control his glycemic responses to food. So I would say it's out there. There's a lot of faff and nonsense as well. Like, it's a capitalist society. There's always going to be someone trying to take advantage of you. But for the most part, there are companies out there doing incredibly exciting science that is changing the world. It takes a long time to get things into the clinical sector. But even without that, I would argue that there are very few people out there who are interested in their gut, interested in their diet, that haven't heard about the microbiome. So from a zeitgeist perspective, a paradigm shift, as Kuhn would have said, we're dealing with something where the society now accepts the fact that they're not just feeding themselves, they're feeding bacteria in their intestine. And if you do that, it has a role. So while we wait for clinical science to catch up, which can take decades, right? I think we're at a space where people are accepting that there are information they can use that could be beneficial in helping them to live a healthier life, right? Eat more fiber, eat the rainbow. And now we know why it's important.
Right. Yeah, I mean, I think it's just such an exciting time right now with the pace of translational research that's coming out in the literature. I feel like we're on the cusp of a lot of stuff happening really fast.
I know, I've been saying that for 10 years though,
but like it's happening, right? Like Day 2 is happening. I just saw they had a study out where they proved that their personalized diet was actually better at reducing insulin levels compared to a Mediterranean diet, which is sort of the standard of care of like a good diet. So it's happening. It's happening.
Yeah, it's funny how people, the one solution fits all is great for the average. Unfortunately, the funny thing about an average is there's a lot of people above it and below it, and and it's those people that miss out. And so, with all of these things, if the microbiome can do anything, it can help stratify patient populations and participants into groups that we can then use to target more effective and personalized therapies.
Great. Okay, well, now we will get to our lovely audience's questions. So first off, I see we have a question about the American Gut Project. And essentially it's similar to what I asked about the Earth Microbiome Project. What is your biggest takeaway from that project?
Yes, this is formulated by Rob Knight in 2011, launched in 2012 with my help and a bunch of other people. And it's still trundling along. We're about to relaunch the American Gut Project next year. And we are, because we wanted to take all the information, all the knowledge that we've gained from the 30 or 40,000 people that have already donated to the project, right? People donate to the project and they have the microbiome sequence so that the microbiome information can go into a glorious free database, which we can use then to identify how the microbiome associates with traits in large populations. And in fact, many of the clinical companies out there utilize the American Gut Project to help understand how their therapies might work. So my biggest takeaway from it is that crowdsourcing science can be successful. The biggest scientific takeaway from it is that if you eat more than 30 species of plant a week, you'll have a very diverse microbiome and metabolome, the chemicals released by those bacteria. And I've taken that to heart, right? A little bit of fermented food and a lot of fiber and diversity of food, right? Eat as many different things as you can and don't try and eat the same thing all the time, and that has the biggest impact for health.
Yep. And one of the big takeaways from me for both the Earth Microbiome Project and American Gut is just the huge value in making data free to everybody, whether it's industry and science. And I know my lab has used those data sets over and over again as sort of comparison cohorts or validation cohorts. So it just sort of accelerates science, generally speaking, to have those kinds of big data sets.
That's exactly what we wanted it to be, right? It was a way of taking our little data sets and comparing them to something big so we can see, we found this bacteria, it's associated with, eye allergies or whatever. Is it really, how is the thing distributed in the population at large? It's just an unfortunate, we've been trying to fill the gaps to make it more equitable and socially responsible and socially, representative. And that's hard, man. That's hard because there are a lot of people out there that we want to capture who don't have the money to donate to science. So we rely on other people to donate for them. So if that's something you're interested in, you want to get, a more diverse representational cohort, find people who'd be willing to donate to support other people getting involved, right?
Yep, great point. Next question from the audience is, what do you think is one of the biggest misconceptions about the human microbiome?
But diversity is really important. Right, I know I've been pushing this for a while. Sometimes diversity, a highly diverse microbiome, can be one that's unstable, and I know you've been interested in that idea for a long time. And an unstable microbiome may not be one that's helping you. A low diversity microbiome is often one that's either bad or good. But bad or good is binary, right? You can have a good microbiome that's low diversity, and you can have a bad microbiome that's low diversity. I think the key concept comes from a highly diverse microbiome is probably more robust to change, right? And therefore, if it's highly diverse in a healthy state, that's a good thing. But it's just relying on diversity as a measure of benefit or health, I think, is one of the most annoying and almost hypocritical scientific failures, right? It's too simple. And, a lot of that stems from doctors wanting really, really simple metrics they can use to understand what the hell's going on. Because when you're used to, the chemical in the blood is X, or, your H1BCA2 score, whatever is Y, that's easy to understand. The microbiome is not easy to understand. It's incredibly complicated. And even the ecologists studying it are really only starting to get to grips with it in the last few years.
Yeah, no, I couldn't agree more with those points. I mean, we had a paper a year or two ago. Tom Wolmansky was the first author on it, where we observed that people who were constipated had really high alpha diversity.
Yeah,
and is
right.
good constipated? I don't know, probably not. I remember I stood up at a conference like year or two years ago, right before the pandemic, and I said that essentially that, well, it's context dependent. Maybe it's good, maybe it's bad, depends on the context. And it was like I farted. Like everyone hated me. Yeah.
They look at you like, well, that doesn't help me. I mean, the person I think who's doing some of the most interesting work in this space at the moment is Mariana Bindlos. She's a phenomenal professor, and her work is really trying to pinpoint exactly what we mean by dysbiotic, right? When someone says you have dysbiosis, what do you mean? It doesn't mean you have low diversity. That's bullshit, right? Pardon my French or English. It's actually completely related to very, very specific breakdowns in the chemical communication between the bacteria and the body. And she's uncovering those and making them available and translatable for doctors, right? And that's where we should be working at, really getting down to the mechanistic brass tacks of what we mean by these statements.
Yep. Next question from the audience kind of goes towards translation. What types of microbiome products or technologies can we expect in the coming years? Things that we don't see yet, but we might see in like the next couple of years.
Well, a few things I'm super interested in. Some of the work coming out of David Mills' lab up at UC Davis and spun out into a number of companies, but dealing with the fact that adding certain types of bacteria into the guts of infants that are breastfeeding can have a massive impact upon their immune training. So Bifidobacterium longum infantis as a probiotic therapy for improving childhood immune health. I think that's a really low-hanging fruit option, but it's a phenomenally powerful option for changing the face of chronic allergic disease in our country. I'm super excited about the field of psychobiotics, as John Cryan would call them, using bacterial probiotics that produce neurotransmitters to have an impact upon mental health. And we actually formed a company, and I do have stocks and shares in that company called Holobiome, that is trying to commercialize those kinds of products. So I'm very excited about that work. And I got a really, really cool new graduate student called Marisol Dothard, who's spearheading that work in our labs at the moment and doing some phenomenal work with rats and depression, anxiety. So I'm really excited about that stuff. I think there's also some incredibly exciting stuff coming out on trying to understand diet and immune health more generally as we age, right? The implications for what it means to maintain a healthy gut, to maintain a healthy body, to prolong a healthy longevity, right? And I'm not there yet, hopefully, but I can definitely feel my 40-year-old bones getting achy. So I'm hoping that we can get that sorted before I hit 70.
Nice. And I also happen to know you are a co-founder or a founder of BiomeSense, right? But I guess that's more targeting research, right? Not necessarily consumers, but
well, it will be consumers at some point. So this is this is like an automated stool sampler, right? So you stick it in your bathroom, drop a piece of toilet tissue in the top, and it extracts all the DNA and RNA and nucleic acids. And so every stool sample we can capture, right? Every single time you do it. And your work, this is inspired by your work, which you and Eric, and demonstrating that we can't just take one sample and compare it to a population and expect to find a marker. That marker of a disease may be present in your poop one day and disappear the next. And if we pick the wrong day, then you don't show up in our personal assessment of whether you have that marker. That's super important to us. So yeah, it is based on research now. But we hope that product can be incredibly powerful for helping to stratify patient populations to improve the outcomes of therapies. Billions, tens of billions of dollars are wasted on clinical trials every year because we can't get the patient stratification right to determine if those drugs are effective. Those drugs probably are effective for a section of people, right? But we can't figure out which people to test. I think the microbiome holds that information, but I think we need to use longitudinal characterization to understand the dynamics to figure out which people would be responders and non-responders. Once we have that information, you can design clinical trials, and that will accelerate drug development. and save money.
Yeah, no, we've been arguing for a few years here at ISB that you should design clinical trials with maybe smaller numbers of people, but with much denser data longitudinally collected. So that if the trial fails, at least you have something to go back to and be like, oh, well, actually, the drug worked in these 20 people, but it didn't work at all in these other 30 people. It's just that it was very heterogeneous, but you could see that when you have the data to look at it. But if you're just doing a simple clinical trial where you're only measuring a couple of things, you totally miss it. Yeah, that's interesting.
Yeah, it's it's one of those things where you're hitting your head against a brick wall of a lot of greybeards who have been doing this their entire lives, and this is the way it's done. And when you try and tell them you're doing it wrong, generally they don't respond very well. But they are, and that's what's annoying.
Jack, we're just too innovative. That's what I keep
telling
myself.
We're so young. We're so young and hip.
So another question from the audience. You mentioned your kids. How has your knowledge, for example, dirt is good, affected your parenting style? What advice would you give to new parents?
Yeah, I know I've got colleagues who have written very similar books who are really, really strict with their kids' diet. I'm not. My kids, it's my youngest son's birthday, and he had pot stickers. That was what he wanted. That's what he got, right? And if he wants mac and cheese, I'll give him mac and cheese as well. But what I try and instill in them is the fundamental understanding, right? And showing them that they can eat healthy. So yeah, we eat a lot of diverse fruits and vegetables on a regular basis. It's always there. It's always available. And that's our privilege in being, in a wealthy position in this world. There are lots of people who don't have access to that kind of dietary thing, and children grow up without access to the food due to food deserts. And when your local grocery stores are 7-Eleven, you're going to suffer some major problems. But I think teaching them about how to live a healthy diet has been key. Also my eldest son has autism, right? And so we played a hell of a lot of role in trying to understand how to regulate the side effects of his neurodiversity, which he doesn't like. There's bits about being autistic which aren't particularly pleasant sometimes, and so managing those aspects for him has been something we played around with a lot. And a lot of that's just diet again, making sure that he has the right kinds of foods, making sure we find which foods make him feel good and which ones make him feel bad, and avoiding the bad ones, right? And then and working out solutions that are peer reviewed and been gone through the appropriate clinical trials, and seeing if he wants to try them. Those kinds of strategies, I think, have played out, in my parenting style in significant ways.
Interesting. Yeah, that last piece of sort of experimenting with foods one at a time and seeing what works. I know a lot of people out there tell me this is what they do themselves. If they, a lot of people have sour guts and issues with their guts, and it's hard the docs don't know what to do. And so my mother has done this in herself of kind of experimenting with what seems to be working, what doesn't work. It takes a long time for people to figure it out, but oftentimes people do come to understand like what foods tend to be good for them, what aren't. And I think science needs to catch up to these N of 1 type trials. I mean, there are designs like crossover trial designs where you can look across a population with these sort of personalized interventions. And use this person as their own control. But I think that just not enough people are doing it, maybe because the graybeards are preventing these kind of trials.
Yeah, definitely the graybeards. We'll blame it on them. I mean, it's just because there's so many variables, right, to try, and it can be overwhelming, right? If you're lactose intolerant, you're probably missing Blauschia, a particular type of bacteria that has, lactase and can degrade lactose, right? So, if you're missing Blauschia from your gut, Rosabardis or Rosaburia as well, you're very unlikely to have a good response to certain types of dairy products. Understanding that's kind of key, right? And you can figure it out from looking at the microbiome now. But for years, people just had to suck it and see. Oh God, every time I had that milkshake, it made me feel bad. And that's where we're at with about 90% of all the problems. But it's compelling. There's another company, I'm on their scientific advisory board, and they're just about to publish a paper where they've demonstrated that you can accurately predict what type of IBS you have based upon the microbiome from a study of over 400, 500 people, right? So we're getting there. If you know what type of IBS you have, it's possible that you can get targeted treatment to actually help to alleviate the symptoms. So those things are coming down the pipeline.
Cool. Well, the time has really flown by here. I think we're getting close to the top of the hour. We have time for one more audience question, I think, before we wrap up. So I'll choose this one. What do you think about fecal transplants for conditions other than recurrent C. diff, right? There's a lot of trials going on that have been done that are ongoing. Do you see potential there? And also comparing that to like bugs as drugs companies that are trying to create cocktails or individual bugs to treat these different diseases.
Yeah, I think there's a lot of potential, right? And it's just going to take a while for the investment firms to fund the trials to actually get this stuff done. I've seen some really compelling preliminary phase 1 and phase 2A/B. These are FDA level trials that demonstrate quite effectively that you can use FMT and potentially bugs as drugs in these small cocktails for improving outcomes in cancer therapy and oncology. That you can some compelling but not particularly rigorous data coming out of Arizona, Rosa Krauss-Malnick-Brown's stuff, demonstrating that you can alleviate some of the side effects of neurodiversity and autism, and that it could potentially be beneficial. There's some really compelling stuff around treating cardiac diseases, metabolic disorders. So these things are coming through, right? But we need better data. We need more rigorous data. We need time series data, right? Damn the graybeards. We got to get the right data to demonstrate for whom it works and for whom it doesn't work. And if we can have that data, these things could be incredibly powerful.
Exactly. I mean, I think one of the keys to that, it's hard to create one pill for everybody, and that's just as true for a bug as it is for a drug. And for a lot of these interventions, there are going to be subsets of the population that just respond wonderfully to that intervention, and other subsets that do not. And so, being good at understanding the heterogeneity and being able to partition people in a personalized fashion to deploy these treatments is, I think that's the future, but it's going to take some time to get there.
And I'll make a pitch, one last pitch, just to say we need more diversity in our cohorts. We're just about managed now to get 50% men, 50% women. We need more trans people embedded in our studies. We need more ethnic diversity. We need more neurodiversity. We need to embrace the fact that we're not all, apart from what this video shows, middle-aged white guys with long hair. There is a world out there, and it needs to be included in our clinical studies in order to demonstrate our support for everybody.
Totally. All right. Well, on that note, this has been a wonderful conversation, a wonderful evening. Thanks so much, Jack.